The biggest danger facing beachgoers may not have fins or sharp teeth or stinging tentacles. There's also a danger in what you can't see. Microbes that can give you stomach cramps, fever, and nausea may also be swimming in the water with you.
There are pathogens in the ocean that are native to the ocean and to estuaries and to lakes and to rivers. They're naturally found there.
But there are other, completely different pathogens that enter the ocean from the land.
So we have all of this infrastructure, sewage, septic systems, pets, livestock, all of these things that are basically now right on the coast and no longer is dilution the solution to our pollution. We've got to consider the feedback loop. So we're not only impacting our environment, but we're also the recipients of those adverse impacts.
The state tests 240 coastal beaches for microbial contaminants between April 1st and October 31st. North Carolina always ranks high in beach water quality. Still, in 2012, swimming advisories were issued for sections of 33 beaches in Beaufort, Brunswick, Carteret, Dare, New Hanover, and Pamlico counties. That's about 1% of the state's beaches. Most of the closings lasted for about a week.
We're testing for bacteria, so the filter simply has to be small enough in the holes that are on the filter to capture the bacteria.
But Dr. Rachel Noble believes the current water test, which requires almost 24 hours to grow bacteria from a water sample in a lab and count it, takes too long.
When you take a water sample at the beach, on a Saturday morning, let's say, they take a water sample at the beach. Even though you're swimming at the beach, the result of that test and the sign that could be placed if that water were contaminated would not be in place until Sunday morning.
Dr. Noble and her team at the University of North Carolina Chapel Hill Institute of Marine Sciences have created a faster water test. It produces results in about 4 hours rather than 24.
The organisms that we test for are the same Enterococcus, E. coli, or fecal coliforms that have been tested for by EPA for decades. The technology is just slightly different.
The test uses what's called quantitative polymerase chain reactions, or qPCR for short.
You can use an array of molecular techniques to basically process the DNA or RNA that might be in your sample that you're looking for. It could be an array of organisms.
It allows scientists to spot DNA from specific types of bacteria and then determine how much of that DNA is present. That's the key, looking for DNA chains tied to specific bacteria that make people sick. Here's how the process works.
We have our sample that was collected at a local beach, and we're going to filter it through a special pore filter, which is 0.45 microns wide. And this process will capture all of the bacteria that's in the water. In this case, we're again looking for the indicator species Enterococci.
Different size filters are used to screen for different bacteria.
I'll pull the filter off of the vacuum and place the filter directly into the tube containing the glass and silica beads.
The cells are then broken apart in this machine. The glass and silica beads help break apart the cells, and that releases their DNA. The sample is then placed in the PCR machine, and the search begins to measure the amount of the particular DNA you are looking for.
You are able to find your target, and then the polymerase chain reaction makes more copies of that target. So it's a series of chemical reactions, and then it's a series of just heating and cooling the sample. And by the time you get to the end of your cycles, essentially, using that machine, you have either been able to quantify the target, or you can find that there was no target actually in your sample.
The continual heating and cooling cycle speeds up the copying of DNA, which allows for a quicker analysis.
And these are those heating and cooling cycles. It basically just repeats itself over and over again. And at some point, you measure the increase in your target DNA, in this case, the Enterococcus DNA.
The faster you see indications of DNA from dangerous bacteria in the sample, it means the more bacteria is present in the water, and the beach should be closed.
I also feel like I'm doing a service to the public by protecting public health, because we can get an answer out sooner. So if I can help develop some tests that will actually help get answers out sooner, better, faster, then that makes me feel better.
There's a whole range of clinical tests that are using the exact same technology. Forensics, for example, all of the DNA technology that's currently used also utilizes polymerase chain reaction. So it's a technology that's been used for decades now. It's not novel or new anymore. And now we're applying it to do some new things in water. We don't want to make people unnecessarily concerned about naturally found bacteria that are doing good things in the marine environment. We want to know more about those ones that are actually potentially making people sick or causing wound infections and causing death.